So continuing with the small intestine, again it's about 20 feet long, 6 meters, all packaged neatly into your abdominal pelvic area, your abdominal area. And then the three regions again: duodenum, the first portion after the stomach, jejunum, here in purple, and then lastly the ileum, which connects with the large intestine. That was at what is referred to as the ileocecal valve that we'll get to. And then the starting of the duodenum begins at the pyloric valve, where it meets with the stomach. It's held in place by the mesentery proper, if you remember that. Let's look at the duodenum, the first portion. It's not that long, only about 10 inches. Right after the stomach, it leads to the jejunum. Internally, histologically, but also structurally, you'll see, or the duodenum has what are called circular folds, these larger folds that you see here, shown here in the diagram, also called the plicae circulares, and they function in increasing surface area. So as the food material, the chyme, percolates through, works its way through, it has more surface contact area with the circular folds, and also helps slow down the movement of the food through the duodenum of the small intestine, and that gives the digestive enzymes time to work on breaking down the food nutrient molecules. Excuse me. Also into the duodenum, it receives materials from the liver, being bile, and then from the pancreas, the pancreatic enzymes, digestive enzymes. So the bile and the pancreatic ducts empty into the duodenum, at what's referred to as the duodenal papilla here. So they both merge, as we'll see, the bile duct coming off of the liver and the gallbladder, bile duct here, and then the pancreatic duct, usually the main pancreatic duct, they merge together at the hepatopancreatic ampulla. So hepato for liver, pancreatic for pancreas, and then their material is secreted into the duodenum of the small intestine through the duodenal papilla. So, bile that's secreted by the liver, stored by the gallbladder, and the bile that enters into the small intestine. The function of bile is to emulsify the lipids, the fats. What emulsify means is break it into smaller droplets, and that increases surface area so that the enzymes from the pancreas can more efficiently break down the lipids into their monomers. And then the pancreatic juice that's produced by the pancreas, that's also secreted into the duodenum of the small intestine, includes digestive enzymes for all of the biological molecules. Proteases to break down proteins, carbohydrates to break down carbohydrates, lipases to break down lipids, even nucleases to break down nucleic acids. And then also the pancreatic juice is basic or alkaline, and that'll increase the pH to close to pH 7, because if you remember, in the stomach, the gastric juices in the stomach are acidic. So the chyme that enters in the duodenum is also acidic, and if it wasn't balanced, it could actually damage the lining of the duodenum, causing problems like ulcers. Okay, moving to the jejunum here in, I guess, purple. It's about 8 feet long, so it's fairly long. Bulk of the digestive breakdown occurs there, and the nutrient absorption. Then lastly, the ileum is about 12 feet long, fairly long, and then some nutrient absorption is occurring there as well, and some breakdown of food nutrients as well. And then here you can see the ileocecal valve, which is the junction of the ileum, the last portion of the small intestine, with the large intestine. There you go. So histology, small intestine, simple cuboidal epithelium, like with the stomach. A large surface area for absorption. So the small intestine has the modifications of the circular folds, and then villi, which are smaller finger-like extensions. They give the appearance of an opened small intestine region, kind of the appearance of kind of like a velvety appearance, and that's the villi, which you can see with the naked eye. And then lining the villi is the epithelium, simple columnar epithelium, and the epithelial cells themselves have extensions, microvilli, which you cannot see without a microscope. But the microvilli on the individual cells of the simple cuboidal epithelium, the villi, the finger-like extensions of the wall of the small intestine, in addition to the large circular folds, relatively large, all act to increase surface area, to slow down the passage of food, but also to increase surface area for absorption of nutrients. The circular folds are also called the plicae circularis. So in this picture, the large structures, so the whole thing is this cross-section of the small intestine, and then these large regions, those are the circular folds, plicae circularis, and the little extensions, the fuzzy part that makes it look like a velvety or like a shag carpet, those are villi that you can see with the naked eye. And then on the villi, the epithelial covering, which you can't see because they're too small, are the microvilli. Here's just another picture to show a zoomed in area of the villi. Again, huge surface area, because the small intestine has these modifications, because that's where the absorption of food nutrients is going to be occurring. The finger-like projections of the villi, part of the mucosal lining. Within the villi, there are blood vessels and lacteals. And lacteals are a component of, notice they're green in color, of the lymphatic system. So the function of the lacteals is to absorb lipids from your food, and then lipids are transported as lymph, or as part of the lymph, through the lymphatic system, and then enters into the venous blood supply later. And then what enters into the blood supply here in the villi are the other nutrients, proteins, amino acids, the breakdowns of carbohydrates, even the breakdown material of nucleic acids. And then that goes directly into the bloodstream, then transported towards the liver. The microvilli form what's called the brush border on the individual cells. As you can see here, this would be one cell, possibly two. That looks like a cell boundary. But those extensions of the microvilli on individual cells, again, magnification is here 18,000. So again, you'll only be able to see those under magnification, so microvilli. On a typical microscope slide, like for lab, these long finger-like extensions, those are individual villi. Villus is singular. And you can see simple cuboidal epithelium, underlying connective tissue. Nutrient absorption. Okay, so the proteins breaking down to amino acids, and they're absorbed across the epithelial wall, transported down into the capillaries. And then so the breakdown of proteins, amino acids, nutrients-wise, enter directly into the blood supply. Same thing with the carbohydrate breakdown materials, the monosaccharides, straight into the capillaries, across the epithelial lining, and then transported into the blood supply there, the capillaries, within the villi. And again, the lipids then transported all the way across the epithelial lining, but they enter into the lacteals of the lymphatic system. Okay, so after all that nutrient absorption and breakdown of foodstuffs, the remnant material enters into the large intestine, or the large bowel. It's about 5 feet long. Principal functions: absorbing water, ions, salts, a little bit of nutrients, and then helping compact the material so the chyme that enters gets compacted, dried out a bit, and turned into feces, fecal material. And then that's what's defecated out of the body. nutrient absorption in the sense of vitamins. A fair amount of vitamins are absorbed, and this is a result of the symbiotic association we have with bacteria that naturally inhabit our large intestines. So those symbiotic bacteria we have in our large intestines are an important part of our health, those organisms, and an essential part of having us maintain a well-functioning digestive system that maximizes absorption of nutrients and vitamins. Excuse me. So then also, I guess the last function here of the large intestine is to store the fecal material, and then you'll get the sensation to defecate, and at which point you should defecation. Okay. The regions. We have the cecum, which is this little inferior part of this portion of the large intestine, just below, inferior to the junction with the ileum and the large intestine, the ileocecal valve. So this pouch down here, that's the cecum, and then there's the appendix coming off of it. Okay. An appendix is part of the lymphatic system, and if it gets blocked, which is possible because it's fairly thin, diameter-wise, very narrow diameter, can lead to appendicitis. And if anyone has had an appendicitis, you know how painful that is and how dangerous that is, and has to get removed, cleaned up promptly. From the cecum, we have the colon, which is between the cecum, technically between the cecum and the rectum, that portion of the large intestine. So we have the ascending colon, meaning the portion of the colon that ascends, moves up superiorly towards the liver. Then there's this 90-degree turn at the hepatic flexure, or the right colic flexure. And then that transitions to this portion that cuts across, the transverse colon. That cuts across from our right side to our left side, towards where the spleen is. Hence, this turn is the splenic flexure, or the left colic flexure. And then this part that goes down is the descending colon. And that transitions to the sigmoid colon. Notice kind of an S-shaped portion here. So then down inferiorly into the pelvic cavity, we have the sigmoid colon, which extends into the pelvic cavity. Then the rectum, where fecal material is stored. And then when this fills to a certain amount, you get the urge to defecate. And then once the defecation urge, or the defecation sensation is felt, the stored fecal material moves down into the anal canal. And then if you're ready to defecate, then out through the anus into the environment. Hopefully into like a toilet or something like that. And then the anus has two sphincters, an internal and external anal sphincters. So the internal one is the deeper inner one, okay? And that is under unconscious control. Autonomic nervous system will start to relax that musculature of the internal anal sphincter, allowing the fecal material to start to descend further into anal canal. But we have the external anal sphincter, which is skeletal muscle under conscious control, and that we can contract, hence closing the anus, meaning holding your fecal material, stopping defecation. And usually we do that because we're not always sitting on the toilet just waiting to defecate. And you can delay the defecation reflex for a while until you get yourself to a bathroom. All right? So internal anal sphincter is involuntary control. External anal sphincter under voluntary control. Yes. Okay, so looking at the histology, again, simple cuboidal epithelium. Like much of the digestive tract, there's no villi. So that's one change from the small intestine. You notice that there is kind of these puffy outpocketings.